Hello, welcome back to Sleep Science. Tonight we are going to talk about something that happened today, probably without you thinking about it for even a moment. You ate something that grew in the ground. Maybe it was an apple, maybe it was a handful of almonds, or a piece of bread made from wheat, or a tomato sliced onto a plate at lunch. Maybe you barely noticed it. Maybe you were distracted, thinking about something else entirely, eating almost on autopilot. And right now, somewhere inside you, something from that food is crossing a barrier it has crossed 1 billion times before. Not in you specifically, in humans. In every human who has eaten what grew from the ground, which is every human who has ever lived. The molecules are moving from gut to blood to cell, and they are doing things that took scientists decades to name, and some things that have still not been fully named, and some things that may never be fully named, because the chemistry is so layered, so tangled, so ancient, that our instruments barely know where to begin. The strange part, though, is this. The plants were not making any of it for us. Not originally. Not for humans. Not with anything like intention. They were just trying to survive. Growing in the dark. Pushing roots down through soil that was itself alive in ways we are only beginning to understand. Making chemicals because chemicals were the only tools available to something that cannot move, cannot run, cannot hide from whatever is eating it, or crowding it, or shading it from the light it needs. And then something happened. Slowly. So slowly that no single generation could have noticed. The chemistry of the ground and the chemistry of the animal body began to fit. Not because anyone planned it. Not because the earth cares about us or knows about us in any meaningful sense. But because fit was what survived. Mismatch died. Agreement persisted. And over millions of years, something that started as a plant defending itself became something a human body could not easily do without. That is the thing I want to talk about tonight. The quiet conversation that has been going on between the soil and the body for longer than the human species has existed. A conversation without words, without intention, without anyone on either side knowing it was happening. Just chemistry, patient as stone, working itself out across geological time. And the remarkable, almost unreasonable result. That what grows in the ground fits what lives above it, with a precision that still surprises scientists who have spent their entire careers studying exactly this. Let's start at the beginning, which is also the ground. Reach down and pick up a handful of soil. Not sand, not clay, not the pale dead stuff from a construction site. Real soil. Dark, slightly damp, smelling of something old and alive. That smell, actually, is worth stopping at. There is a compound called geosmin. It is produced by bacteria, specifically a group called actinomycetes, which live in topsoil almost everywhere on Earth. When soil gets wet after being dry, these bacteria release geosmin into the air. And that is exactly what you are smelling when rain hits dry ground. That ancient, unmistakable smell. Humans are extraordinarily sensitive to it. Extraordinarily sensitive as in, we can detect it at concentrations of a few parts per trillion. That is a sensitivity that rivals a bloodhound on a scent. Which raises an obvious question. Why? Why would the human nose be tuned so precisely to the smell of bacteria in wet soil? The most persuasive answer is that our ancestors needed to find water. In a landscape before maps, before rivers marked on paper, the smell of rain hitting dry ground was information. Life-saving information. So the ones who could smell it from farther away, who stopped and turned their heads toward it, survived at slightly higher rates than the ones who could not. And over enough generations, that became the nose you have now, which is already a version of the thing I want to tell you about tonight. This sensitivity, this tuning, this fit between a living body and something in the ground. It started before we were even quite human. It runs deeper than civilization, deeper than agriculture, deeper than the first planted seed. But the story of plants and the body goes so much further than one smell. A plant is a chemical factory. This is not a metaphor. This is the actual thing. Not a factory that makes one thing. A factory that runs hundreds of processes simultaneously, responding to conditions in real time, manufacturing compounds of extraordinary complexity from basic raw materials: sunlight, water, carbon dioxide, and whatever minerals it can pull from the soil through its roots. A single plant can contain thousands of distinct chemical compounds, not dozens, thousands. Some of them are what we call primary metabolites. These are the fundamental building blocks, the sugars and amino acids and fats that the plant needs to run its own body, to grow, to reproduce, to stay alive. This is the basic chemistry of life, shared between plants and animals in ways that are so fundamental they show up in everything from bacteria to blue whales. But then there are the secondary metabolites. These are the interesting ones. Secondary metabolites are the chemicals a plant makes not just to run its body, but to navigate the world, to protect itself, to communicate, to attract some things and repel others, to survive fire, flood, drought, frost, disease, insects, fungi, grazing animals, ultraviolet radiation from the sun. The plant cannot move. It cannot fight. It cannot flee. Everything it does to deal with a hostile world, it does through chemistry. And it has had a very, very long time to get good at chemistry. The first plants on land appeared roughly 470 million years ago. The ancestors of the flowering plants we eat today started diversifying around 130 million years ago, though the lineages are older still. By any measure, these are organisms that have been running biochemical experiments under real-world conditions for longer than the human mind can meaningfully imagine. When a compound works, it persists. When it doesn't, that branch of the family tree eventually stops branching. The result, over geological time, is an extraordinary pharmacopoeia. Compounds tested by reality across millions of generations. Compounds that solved specific problems in specific environments. Compounds that, when you look at them closely, turn out to match the biology of the animals in those environments with a precision that keeps surprising people who are paid to not be surprised. That is not an accident. It is coevolution. Coevolution is what happens when two living things shape each other across time. Not one shaping the other, both, simultaneously, changing in response to each other, neither one leading, neither one in control, just two sets of chemistry in an arms race, or a collaboration, or something in between, across enough time that the result looks almost like planning. The classic example is the flower and the bee. The flower needs to get its pollen to another flower of the same species. It cannot move, so it makes something attractive, something colorful, something with a scent, something with a reward, which is nectar. And the bee, drawn to all of this, does the work the flower cannot do itself, carrying pollen from one bloom to another, being paid in sweetness for work the flower needed done. But the bee did not go looking for work. The bee went looking for food. It was already visiting flowers before that particular flower evolved those particular colors and that particular scent. The flower changed in response to what the bee was drawn to. The bee changed, over generations, in response to what was available in flowers. Each one, slowly, became more precisely suited to the other. Neither one of them understanding what was happening. The result is that certain bees and certain flowers now fit each other with a specificity that still startles entomologists. A tube exactly the right length for a particular bee's tongue. A scent that only activates the receptors of a particular bee's nose. The flower not wasting its nectar on visitors who won't pollinate it. The bee not wasting its time on flowers that won't feed it. An efficiency built not by engineering, but by time. Now, apply that same logic to fruit. Fruit is, at its core, a seed delivery system. A plant needs its seeds to travel. Seeds that fall straight down under the parent plant don't do well. They compete with the parent for the same light, the same water, the same soil nutrients. Better for the seeds to go somewhere else, somewhere new. So how does a plant that cannot move get its seeds to travel? The same way the flower gets its pollen moved. You offer a reward to something that can move. You make the seeds valuable to carry. Not the seeds themselves, which you want the animal to deposit, ideally in a new location, ideally in a pile of fertilizing animal waste. You make the thing around the seeds valuable. You make fruit. And what is the most effective reward for a traveling animal? Calories, energy, sweetness. Sugar is energy. An animal that eats sugar gets energy. Energy is the thing all animals are ultimately trying to secure. So a fruit that tastes sweet will be eaten. Its seeds will be carried away and deposited somewhere new. The plant that makes sweeter, more attractive fruit has its seeds dispersed more widely. Its offspring start in more varied conditions. More of them survive. The fruit eaters, meanwhile, are finding more food in the sweet fruits. They thrive. They are drawn back to the same plants. The plants are selected for sweeter fruit. The animals are selected for stronger preference for sweetness. Both are changing. Both are becoming more precisely fitted to each other. Over millions of years, what you get is this: animals with a deep, powerful craving for sweetness, and fruit with a precise ability to satisfy that craving. And when you eat a ripe peach in August, that craving and that satisfaction, they are not modern inventions. They are not agricultural products. They are the result of a conversation that started before the first human ancestor stood upright. You are tasting the end product of something that has been refined across more generations than we can count. But sweetness is only one part of the story. Because while the fruit is sweet on the outside, drawing animals in, protecting the seeds inside, the plant is also quietly adding something else. Phytonutrients. The word just means nutrients from plants, but that translation doesn't really catch what they are. They are not the basic nutrients, not the carbohydrates, not the proteins, not the fats. Those are the primary metabolites, the universal building blocks. Phytonutrients are the secondary metabolites, the thousands of distinct compounds the plant makes to deal with the world. And here is where it gets interesting. A lot of these compounds, these secondary metabolites that the plant made for its own purposes, turn out to do things in the human body. Very specific things. Things that the human body seems, in some cases, almost designed to receive. The human body has receptors for compounds that only exist in plants. Not just general receptors that happen to respond to plant chemicals. Specific receptors that are so well matched to specific phytonutrients that researchers initially thought they must be responding to something the body produced itself. This is the quiet astonishment at the center of this whole story. When your body receives a particular molecule from a plant, the docking is not approximate. It is precise. Like a key and a lock. A lock in your cells that has been waiting, across evolutionary time, for a key that grows in the ground. Let's talk about the walnut. Not because it is the most dramatic example, though it might be the most visually striking one, but because it is a good place to start. A walnut looks like a brain. No, really. Look at one closely. The whole nut, still in its shell, is roughly spherical, like a skull. Crack it open, and what you find inside has two distinct hemispheres connected by a central ridge. The surface is convoluted, folded, wrinkled in a pattern that genuinely resembles the surface of the cerebral cortex. There is a thin, papery membrane between the two halves, almost like the dura mater, the membrane that separates the two hemispheres of the actual human brain. People noticed this a long time ago. For centuries, actually. There is a whole tradition, mostly medieval, called the doctrine of signatures, which I will come back to properly later, that said plants which resemble body parts heal those body parts. Walnuts, by this logic, were for the brain. Modern science largely dismissed this as folk superstition, which is reasonable. Most of it is. Resemblance is not mechanism. Just because a kidney bean looks like a kidney doesn't automatically mean it heals kidneys. But then modern science started studying walnuts seriously, and things got complicated. Walnuts are one of the richest plant sources of a type of fatty acid called alpha-linolenic acid, which the body uses to make EPA and DHA, which are omega-3 fatty acids, which are the primary structural components of brain cell membranes. The brain, roughly 60% of its dry weight, is fat. Not any fat, specific fats. And DHA is so central to brain structure and function that a developing human brain, in the womb and in infancy, requires a constant supply of it. A shortage during development has measurable, lasting effects on cognition. These omega-3 fatty acids also regulate neuroinflammation. Inflammation in the brain is linked to depression, to cognitive decline, to a range of conditions that become more likely as the brain ages and the dietary supply of these fatty acids falls short. There is genuine research, good research, careful research, suggesting that the omega-3 acids from walnuts and other sources do something real for the human brain. Something inside the walnut that science took decades to properly identify and name turns out to be exactly what the brain uses to build itself and to stay clear and working. Which is a coincidence, or it is not. Here is the straightforward explanation, the one that does not require any mysticism. Animals that ate walnuts got more of what their brains needed. Their brains worked better. They found food more effectively, solved problems more effectively, survived more effectively. They passed this trait on. Over enough generations, animals in walnut-growing environments had brains that were, in some sense, tuned to the nutrients walnuts provided. Meanwhile, the walnut was making its omega-3 acids for its own reasons, because plant cell membranes also need these fats, because these fats help the walnut survive cold temperatures, because the chemistry had been useful for other reasons entirely, before any animal came along and started eating it. Two separate systems, two separate needs, a coincidence of chemistry that happened to line up. And then, because it lined up, the lining up deepened. Both sides becoming slightly more fitted to the other, over millions of years. That is coevolution, not planned, not mystical, just relentless, patient fitting. Now let's go underground for a moment, because this story cannot be told properly without the soil. A handful of soil, real living soil, contains more individual living organisms than there are people on the surface of the earth. Not more species, more individuals. Billions in a single gram. Bacteria, archaea, fungi, protozoa, nematodes, mites, springtails, earthworms, countless others. A community of life so dense and so intricate that soil scientists sometimes say that we know more about the stars than we know about what is happening beneath our feet. These organisms are not just passengers, they are the engine of the whole system. The bacteria convert atmospheric nitrogen into forms that plants can absorb. Without this, plants could not make proteins, could not make DNA, could not grow. The fungi break down dead organic matter into the mineral components plants need and do something else besides, something stranger and more interesting that I will come to in a moment. The protozoa eat the bacteria and, in doing so, release nutrients in forms that are more available to plant roots. The earthworms move through the soil, aerating it, mixing it, concentrating nutrients in their castings. The whole thing is a metabolism, a vast, distributed metabolism that has been running since before there were plants to feed, since before there was oxygen in the atmosphere, since before the oceans had fish in them. Life processing life, cycling the same atoms through different forms, the same carbon that was once a dinosaur, now in a blade of grass, now in your lungs. And the plants are not passive recipients of all this. They are participants. A plant sends somewhere between 10 and 40% of the sugars it makes through photosynthesis down through its roots and into the soil. On purpose, biologically speaking, not by accident. The plant is feeding the bacteria and the fungi around its roots, selecting for the ones it finds useful, shaping the community living in its immediate vicinity. In return, the bacteria and fungi provide the plant with minerals it couldn't otherwise access, with protection from pathogens, with signals about the state of the environment it's growing in. This exchange is called the rhizosphere, the zone around a plant's roots where the chemistry between plant and soil is most active. And what happens there is genuinely astonishing. The bacteria in the rhizosphere produce compounds that the plant responds to. They produce enzymes that change the chemistry of the soil around the roots. The plant releases chemicals that change the behavior of the bacteria. There are signals being sent in both directions. Not words, not intentions, just chemistry. But chemistry so specific that scientists use the word communication to describe it. Not loosely, but technically, because what is happening fits the definition of information transfer between separate organisms. And some of this communication directly affects what the plant makes. A plant growing in soil rich in certain bacteria produces different secondary metabolites than the same plant growing in sterile soil. The soil community, in other words, affects the chemistry the plant puts into its fruit, its leaves, its roots. The phytonutrients you get from a tomato grown in living soil are not the same as the phytonutrients from a tomato grown in a hydroponic solution of basic minerals. The soil is not just growing the plant. It is, in some sense, shaping the medicine that plant makes. The tomato. In the 1970s, a researcher named Wilhelm Schurk, or it might have been someone at a similar lab around that time, I don't remember the exact origin of this particular line of research, began looking at a red pigment in tomatoes, a carotenoid, a compound the tomato makes to attract birds and other animals to eat its fruit, which is the same reason it is red, the color being part of the advertisement. The compound was lycopene. Lycopene is not exactly doing the health work alone in tomatoes. The biology turned out to be a whole cascade of interacting compounds, the lycopene acting alongside other carotenoids and polyphenols in ways that are still being mapped. But lycopene was the one that kept showing up in the research, the one scientists kept circling back to. Here is what they found. The human body absorbs lycopene from tomatoes and deposits it in several specific tissues. The prostate gland in men accumulates lycopene at concentrations higher than almost anywhere else in the body, which immediately raised an obvious question, because the prostate is not a tissue you would expect to be particularly interested in a pigment from a fruit, except that lycopene turns out to have potent antioxidant properties. It neutralizes free radicals, the reactive oxygen molecules that, when left unchecked, damage DNA, damage cell membranes, accumulate in tissues over time in ways that are associated with cancer. And the prostate, for reasons related to its function and metabolism, is a tissue that generates these free radicals at relatively high rates. So the body takes lycopene from tomatoes and concentrates it exactly where antioxidant protection is most needed. This is the kind of thing that makes careful scientists stop and look at each other. It does not prove intention. The tomato did not make lycopene for the human prostate. The tomato made lycopene to be red, to attract birds, to spread its seeds. But the human body, over evolutionary time, developed mechanisms to absorb and concentrate this particular compound in this particular way, presumably because individuals whose bodies did this well had slightly better outcomes, survived at slightly higher rates, passed the trait on. The fit is real. The fit is precise. The fit is not mystical, but it is striking. One more thing about the soil before we move on. There is a particular organism in the soil called Mycobacterium vaccae. It is a bacterium. It lives in healthy, rich topsoil almost everywhere in the world. And researchers studying it discovered something interesting. When mice were exposed to it, either by injection in early studies or by inhalation of soil particles in later ones, they showed measurable changes in brain chemistry. Specifically, increased levels of serotonin, measurable reduction in anxiety-like behavior, immune system modulation. The working hypothesis is that humans have been in intimate contact with this bacterium for hundreds of thousands of years. Bare feet on soil, hands digging in earth, breathing in particles from the ground. The immune system, shaped by this constant contact, developed a kind of response to it that is, effectively, calming. There is a feedback loop between the gut microbiome and the brain that is well established now. Less well established but genuinely being studied is a similar feedback involving soil microbes and their effect on the nervous system, mediated by the immune system, by the compounds these bacteria produce that we absorb through skin and lung and gut. The ground and the body, in other words, are in a conversation that is not just about what we eat. It is about what we touch and breathe and stand on. It is a very old conversation. And somehow that is one of the stranger, quieter thoughts. That the simple act of walking barefoot on real soil, something humans did for essentially the entire existence of the species, and have mostly stopped doing in the last 100 years, was actually pharmacological, was delivering something, a chemistry so specific that it affected the brain's ability to feel calm. The soil knew, not in any way that involves knowing, not in the way a person knows a thing, but in the way a lock and a key know each other. The fit was there. The exchange was happening, long before anyone understood it or measured it or gave it a name. Let's talk about what a plant is actually doing when it makes a berry, because this too is stranger than it looks. Dark berries, your blueberries and blackberries, and dark cherries and elderberries, their color comes from compounds called anthocyanins. These are flavonoids, a class of polyphenol, which is itself a class of phytonutrient. They are made in the plant's outer layers, the skin of the berry, where they do several things simultaneously. They act as sunscreen. The deep purple and blue and red pigments absorb UV radiation before it can damage the plant's DNA. A berry in full summer sun is, in a sense, wearing sunblock. They attract animals. These dark, saturated colors are highly visible to birds, which is the whole point. The berry needs to be seen and eaten. The seeds need to be carried away. They repel the wrong animals. Certain insects, particularly insects the plant does not want boring into its fruit and laying eggs before the seeds are mature, are deterred by anthocyanins. The color that attracts birds, which disperse seeds effectively, deters certain insects, which would ruin the fruit. The same compound doing two jobs at once. And then, in the human body, anthocyanins do something else again. They are absorbed into the bloodstream. They cross the blood-brain barrier. They have been shown in careful studies to accumulate in brain tissue, where they reduce oxidative stress, reduce neuroinflammation, and appear to improve certain aspects of memory and cognitive function. They also interact with endothelial cells, the cells lining blood vessels, in ways that improve vascular flexibility and reduce blood pressure. The cardiovascular benefits of dark berries are not, the research suggests, simply about fiber or vitamins. They are specifically related to anthocyanins, the exact same compounds that the plant made to protect itself from UV radiation and to advertise itself to birds. The human body did not need UV protection in the same way, but it turns out it needed the exact same class of molecules for related reasons, because free radical damage from UV radiation and free radical damage from cellular metabolism are, at the molecular level, the same kind of problem. The same chemistry that solves one problem solves the other. The plants and the animals were solving different problems, but they were solving them with the same chemistry. And the fit that resulted from this accidental overlap got selected for, reinforced, deepened across time. Until you ate a handful of blueberries this morning, perhaps not thinking about any of this, and your blood vessels were quietly better for it, and your brain had slightly more of what it needed to stay clear. The banana is a different kind of story. It is not about antioxidants or pigments or UV protection. The banana story, the part of it that I find quietly remarkable, is about a mineral: potassium. A banana contains about 422 milligrams of potassium in an average fruit. That is, in terms of dietary potassium, significant. And potassium matters enormously to the human body because it is one of the primary electrolytes governing electrical activity across cell membranes. Every heartbeat is an electrical event, a wave of electrochemical activity that moves through specialized cells in the heart in a precise sequence, causing the muscle to contract in the right order, at the right time, pumping blood. Potassium, along with sodium and calcium, is what makes this electrical activity possible. The gradient between potassium inside the cell and sodium outside the cell is what creates the electrical potential, the charge, that allows the cell to fire. If that gradient collapses, the electrical activity fails. This is what happens in a potassium deficiency. The heart's electrical rhythms become unstable. Arrhythmias develop. In severe cases, the heart stops. The human kidneys are extraordinarily precise regulators of potassium. They monitor blood potassium levels constantly and excrete exactly as much as needs to be lost and retain exactly as much as needs to be kept. The system is finely tuned, but it depends on input, on getting enough potassium from food to maintain the concentration the heart and nerves need. And the potassium in a banana is not incidental. Plants need potassium for their own reasons. It is an essential nutrient for plant growth, for activating enzymes, for regulating water, for photosynthesis. The banana accumulates it. We eat the banana. The potassium moves from soil to plant, to fruit, to blood, to the electrical system that keeps your heart in rhythm. The soil and the heart, connected by chemistry, through a yellow fruit that grew in a tropical country and traveled to wherever you ate it, carrying something your heart specifically needs. Now I want to come back to the soil microbiome, because I said something about mycorrhizal fungi earlier and then moved on too quickly, and it deserves more time. Mycorrhizal fungi are, when you look at them honestly, one of the strangest stories in biology. These fungi form symbiotic relationships with plant roots. Not parasitic, symbiotic. They physically grow into the root tissue. In some cases, forming structures inside the root cells themselves called arbuscules, which is where the actual exchange of nutrients happens. The fungal network then extends outward from the root into the soil, in some cases extending the effective root surface area of a single plant by a factor of 100 or 1,000. Fungal hyphae, which are the individual filaments of fungal growth, are far finer than root cells. They can penetrate into soil pores and reach mineral deposits that root cells physically cannot access. The fungi do the mining. They bring back phosphorus, nitrogen, zinc, copper, other minerals. They deliver them to the plant. In exchange, the plant gives the fungi some of the sugars it produces through photosynthesis. This relationship is ancient. We find fossilized evidence of mycorrhizal associations going back 450 million years to the very first land plants. Which means the first plants to colonize land were doing so in partnership with fungi. The partnership may actually have made the colonization of land possible. But here is the part that makes it stranger. This fungal network does not just connect one plant to one fungus. It connects multiple plants through a single fungal network. The hyphae of a single fungal organism can extend through a forest, connecting dozens or hundreds of individual trees. And through this network, nutrients move. If a tree is shaded and not producing enough sugar to grow, the fungal network may deliver nutrients to it from trees that are producing a surplus. Not always, not in every case. The mechanisms are complex and still being studied, and there is ongoing debate about how much of this nutrient sharing is deliberate versus incidental. Whether the word sharing even applies in a meaningful sense. But what is clear is that the network exists. The transfers happen. A forest is not a collection of competing individual trees. A forest is, in some meaningful biological sense, a community with a shared underground infrastructure. The line between one tree and another is not as clear as it looks from above. And standing in a forest, breathing air made by trees whose roots are threaded through a fungal web that connects them to the soil and to each other, it is very hard not to feel that something important is happening beneath your feet. Something that has been happening for hundreds of millions of years. Something patient. Something almost incomprehensibly old. Something that has been quietly maintaining the conditions in which life, including human life, is possible. The ground knows something we are still in the process of learning. Citrus, oranges, lemons, limes. The story of citrus and vitamin C is, in its own way, one of the most remarkable accidental experiments in human history. Vitamin C, ascorbic acid, is a molecule the human body cannot make. This is unusual. Most mammals can synthesize it themselves. But at some point in primate evolution, roughly 60 million years ago, the gene responsible for the final step in vitamin C synthesis was disabled by a mutation. A single mutation, and the primates in which this happened survived anyway, because they were getting enough vitamin C from their diet. Because primates live in environments rich in fresh fruit and leafy vegetation, both of which contain vitamin C. In an environment where the molecule is abundant in food, the cost of maintaining the machinery to make it yourself is not repaid by any benefit. So the gene degraded, became a pseudogene, a ghost of a gene that no longer does anything. And now we cannot make it. We are completely dependent on dietary sources, which was fine for millions of years because the diet included plenty of fruit. Until European sailors started taking months-long ocean voyages with no fresh food aboard. And then something terrible happened, regularly, to whole crews. Scurvy. The early symptoms are fatigue, depression, irritability, vague aches. Easy to miss. Easy to attribute to other causes. Then the gums begin to swell and bleed. Then the skin. Old wounds that had healed begin to reopen. Then larger bleeds. And eventually, if the deficiency continues, death. Sailors were dying of scurvy on long voyages for a long time before anyone understood why. The connection was eventually noticed, empirically, before anyone knew about vitamins at all, before the word vitamin existed. Something in fresh fruit and fresh vegetables prevented it. Something that was not present in dried or salted food. The British Navy eventually started issuing lemon juice and later lime juice to sailors, which is where the slang term for British sailors, limeys, comes from. They didn't know what they were giving, they just knew it worked. It took until 1932 for vitamin C to be isolated and identified. What it does in the body is many things. It is essential for collagen synthesis, which is the structural protein that holds tissues together. Without it, the collagen in blood vessels, in skin, in the connective tissue around healing wounds, begins to break down. That is scurvy. The body literally coming apart because it cannot maintain its own structure. It is also a powerful antioxidant. It protects cells from oxidative damage. It is involved in immune function, in iron absorption, in the synthesis of neurotransmitters, including serotonin. The citrus plant makes it for completely different reasons. Ascorbic acid is involved in plant metabolism, in protecting plant cells from oxidative damage during photosynthesis, in building cell walls. The plant makes it for itself. We need it desperately. So desperately that without it, we die. And the orange, the lemon, the lime, they have been providing it to primates, including humans, for roughly 60 million years. Long before there were humans to receive it. Long before the first human ancestor had the genetic mutation that made receiving it necessary. The fruit was already making it. We just became dependent on it. There is something worth sitting with in the folate story. Folate is a B vitamin. It is found in dark leafy greens, particularly spinach and other members of that family, as well as legumes, asparagus, avocado. It is the form of the vitamin as it exists in food. Folic acid is the synthetic form. In the body, folate is involved in a process called one-carbon metabolism, which is a more fundamental biochemical process than its boring name suggests. It is central to DNA synthesis, to the production of new cells, to the repair of damaged DNA. The most well-known application of this is in early pregnancy. A developing embryo is essentially doing nothing but making new cells, dividing and differentiating at a rate that is almost beyond comprehension. The demand for folate during this process is enormous. And if that demand is not met, the results can include neural tube defects, failures in the development of the brain and spine that have lifelong consequences. This is well established enough that many countries now fortify grains and flour with folic acid as a public health measure. The results have been measurable. The incidence of neural tube defects has dropped significantly in countries with folate fortification programs. But what I find interesting is the older part of the story. The dark leafy greens that are the primary dietary sources of folate. They are among the most ancient food plants. Humans and their ancestors were eating them long before agriculture. They grew everywhere humans lived. They are among the most nutritionally dense foods in any traditional diet. And the folate they contain is specifically, precisely what is needed for the thing that makes more humans: reproduction. The growth of new life. The formation of the nervous system in a developing child. A compound made by plants for their own biochemistry, the same carbon metabolism that plants use to copy their own DNA when dividing, turns out to be essential, irreplaceable, for the process of making more humans. The chemistry of plant cell division and the chemistry of human cell division overlap in a way that is so specific, so non-trivial, that removing this compound from the diet has measurable, devastating consequences. That overlap did not happen by accident in the sense of being random. It happened because plants and animals share a deep evolutionary origin. We have common ancestors, somewhere in the very deep past, that used this same one-carbon chemistry for the same basic purpose. The compound and the process are older than the split between the kingdoms of life, older than the distinction between plant and animal. In some sense, folate is not a plant chemical that happens to work in human bodies. It is a life chemical that both plants and humans inherited from a shared ancestor that predates both of them by hundreds of millions of years. The ground is not just growing what the body needs. In some deep sense, the ground and the body are made of the same stuff, came from the same source, are still running in parallel, versions of the same ancient chemistry. And then there is turmeric. Turmeric is a root. It grows in the ground. It has been used in Ayurvedic medicine for thousands of years, which is sometimes offered as evidence of its efficacy and sometimes dismissed as pre-scientific folk belief. The honest answer is that both responses are sometimes appropriate, depending on which traditional claim you are examining. The active compound in turmeric, the one that gives it its intense yellow color, and the one that has attracted the most scientific interest, is curcumin. Curcumin is a polyphenol. It is, like most polyphenols, a secondary metabolite. The turmeric plant makes it, as best as researchers can determine, as an antimicrobial and antifungal defense. A chemical weapon against the pathogens that would otherwise invade and consume the root. In the human body, curcumin has been shown, in very many studies, to have anti-inflammatory properties. The mechanism is specific. It inhibits a molecule called NFKB, which is a protein complex that functions as a kind of master switch for inflammation. NFKB activation turns on the genes that produce inflammatory cytokines, the signaling molecules that set off the inflammatory cascade. Chronic inflammation, NFKB staying activated when it shouldn't, is now implicated in a very long list of conditions: cardiovascular disease, type 2 diabetes, Alzheimer's disease, many cancers. It is not the only cause of these things, but it is a thread that runs through many of them. Curcumin inhibits this switch. Now, the problem with curcumin is that it is not well absorbed on its own. Bioavailability is low. This has caused some researchers to question whether the anti-inflammatory effects seen in cell studies translate into meaningful effects in the human body. But there is something interesting about how turmeric has been traditionally used. In South Asian cooking, in the cuisines where turmeric is most central, it is almost always combined with black pepper. Not just because of flavor, though that is real, but black pepper contains a compound called piperine, which dramatically increases the bioavailability of curcumin by something like 2,000% in some studies. The food tradition got there before the biochemistry. People were combining turmeric and black pepper in cooking long before anyone knew about curcumin or piperine or bioavailability. They knew it worked at the level of lived experience, at the level of generations of observation. The science came later and found that yes, the combination does what the tradition said it did for specific molecular reasons. Which is not mystical. It is just empiricism without the vocabulary. A lot of traditional food knowledge is that. Generations of humans noticing what helped and what didn't, passing that knowledge on, not as chemistry but as practice, as recipe, as ritual. And the chemistry, when it was finally possible to look, finding that the observation was correct. Garlic. Garlic is in many ways the most dramatic example of the plant as chemistry factory principle, because the way it delivers its primary active compound is itself extraordinary. Garlic contains an amino acid called alliin. When a garlic clove is intact, nothing special happens to alliin. But when the clove is crushed or cut, the cellular damage brings alliin into contact with an enzyme called alliinase, which immediately converts alliin into allicin. Allicin is the compound responsible for the sharp, pungent smell of fresh-cut garlic. It is unstable. It breaks down quickly, within hours, into other sulfur-containing compounds. The plant makes it as a defense. When the clove is damaged, the attacker, whether insect, animal, or fungus, is immediately exposed to allicin. The compound has powerful antimicrobial and antifungal properties. It is, functionally, a chemical alarm triggered by injury. In the human body, allicin and its breakdown products have been shown to inhibit bacterial growth. Broad spectrum. They disrupt bacterial cell membranes, interfere with bacterial enzyme systems, work through mechanisms that bacteria have much more difficulty evolving resistance to than they do with conventional antibiotics. There are genuine researchers studying garlic compounds as potential sources of new antimicrobial agents, precisely because the mechanism is so different from anything we currently use. Garlic has also been associated with cardiovascular effects, with modest reductions in blood pressure, with improvements in cholesterol profiles. The mechanisms are still being worked out, but the epidemiological signal is consistent enough to take seriously. What I find strange about garlic is this two-stage delivery system. The alliin, sitting dormant, waiting. The enzyme, separated from it by the architecture of the cell. The allicin only created when the cell is broken. It is almost as if the plant designed a chemical that is only activated by injury. Which is exactly what happened. The plant was under evolutionary pressure to deter things that bite and chew. The chemistry that arose in response is triggered specifically by biting and chewing. The plant is releasing its defense at the moment of attack, not before, not after. And we learned, somehow, to cause exactly that damage ourselves. To crush and cut and break the garlic before we use it, triggering the chemistry, using the plant's defense for our own purposes. The garlic defending itself. Humans learning to use that defense. The chemistry between them finding, as chemistry so often does between plants and animals, an unexpected agreement. I want to stay in the soil a bit longer. There is a researcher named Graham Rook, an immunologist in London, and his work on what he called the old friends hypothesis, which is sometimes still called the hygiene hypothesis, though Rook and others prefer old friends because it is more precise, has a quiet implication that I keep thinking about. The basic idea is this. The human immune system did not evolve in a sterile environment. It evolved in constant contact with microorganisms, with parasites, bacteria, fungi, soil organisms. Millions of years of contact with these things shaped how the immune system develops, what it tolerates, how it calibrates itself. The immune system, Rook and others have argued, actually needs this contact to develop properly. Not to be infected, not to be harmed, but to receive the right signals, to establish the right tolerance, to calibrate the balance between reactivity and tolerance that keeps it working correctly. Remove those signals, place a child in a very clean environment without regular contact with soil organisms and the full range of environmental microbiota, and something in the immune system's development goes wrong. Not dramatically wrong, subtly wrong, in ways that show up later as allergies, asthma, inflammatory bowel conditions, autoimmune disorders, and, intriguingly, anxiety and depression. The immune system, calibrated against the microbial world of the ground, turns its attention inward when that world is absent, becomes reactive to things that are not threats, becomes dysregulated in ways that have downstream effects on every system in the body, including the nervous system. The ground has been calibrating the immune system for the entire existence of the species. The contact between soil and skin, between soil organisms and gut, between the microbiome of the earth and the microbiome of the body, was not incidental. It was necessary. It was shaping us even as we ate what grew from it. And so the question of what the earth grows that the human body needs turns out to be not just about food, not just about phytonutrients and minerals and vitamins. It is about the organisms themselves, about the microbial community of the soil being, in some essential way, a part of the environment the human immune system was designed for and still needs. There is a sense in which the boundary between the inside of the body and the outside of the world was never as clear as we imagined. The body, the skin, the gut, the lung, they are all surfaces where the outside comes in, where the microbiome of the earth encounters the microbiome of the body, and each one shapes the other. The body has been, for its entire evolutionary history, partially made of the ground, partly containing the ground, in a constant, quiet, uninterrupted exchange with it. And the exchange has been so long and so consistent that the body depends on it, not just for food, for the calibration of its own defenses, for the signals that tell the immune system what is friend and what is enemy, for a kind of orientation, a kind of grounding that the body seems to need as surely as it needs air. Let me come back to the specific chemistry of the berries for a moment, because I moved through it quickly and there is a thread I want to follow. The flavonoids in dark berries, the anthocyanins we discussed, are part of a much larger family. The flavonoids as a group include thousands of distinct compounds. Quercetin, which is in apples and onions, resveratrol in grapes and red wine, though the concentrations in wine are lower than often claimed. Catechins, concentrated in green tea. Camphorol in kale and broccoli. Apigenin in parsley and chamomile. Epicatechin in dark chocolate and various berries. These are not all the same thing. They are a family, related by structure, but diverse in function. Each one doing different things in different tissues. Each one the result of a different branch of plant chemistry, evolved for different reasons in different plant lineages. But they share, at the molecular level, a kind of functional similarity. They are all polyphenols, meaning they have a specific structure of phenol rings that gives them their antioxidant properties. And in the human body, they seem to speak, in a sense, a common language with the signaling systems of our cells. Human cells have receptors, enzymes, transport proteins that interact with these flavonoids. Not just one or two, multiple systems, multiple pathways that respond to these plant compounds in specific ways, modulating inflammation, modulating the expression of genes involved in cell survival and stress response, modulating the behavior of the microbiome in the gut. These are not the same pathways the plant used these compounds for. The plant used them for UV protection, for antimicrobial defense, for pigmentation. The human body is using them for regulation, for tuning the system. And what is strange is how specifically human biology seems to be set up to receive and use these compounds. The transport proteins that carry flavonoids across gut cells into the bloodstream, the enzymes in the gut microbiome that transform certain flavonoids into more bioavailable forms, the specific tissues where they accumulate. None of this looks like an accident. All of it looks like the result of deep, patient evolutionary time. The plants making these compounds for their own reasons. The animals in the plant's environment being affected by them. The animals whose biology worked better with them, surviving more, reproducing more, having slightly more of the machinery to absorb and use them. The reciprocal selection pressures building across millions of years until the fit between plant chemistry and animal biology is so precise that it sometimes fools researchers into thinking the compounds must be something the body produces itself. There is a dark-leafed plant that grows at high altitudes, in thin soil on steep slopes, in conditions that most plants avoid. It produces a compound in its leaves specifically because of the UV radiation at altitude. The same compound turns out, in the human body, to modulate the same pathways that UV radiation disrupts in human skin cells. The plant making its sunscreen. The human body using that same chemistry for the same general purpose, applied to a different tissue in a different context. These convergences, and there are hundreds of them, are not individually mysterious. Each one has a straightforward evolutionary explanation. But standing back and seeing them together, the feeling that is produced is something like looking at a very old piece of craftsmanship and realizing that the joints are perfect not because someone planned it perfectly, but because the wood and the method have been worked together for so long that they have shaped each other into a fit that looks intentional. The ground grew the thing. The body learned to receive it. Both of them changed in the process, across geological time, until what the ground grows and what the body needs are so deeply matched that you can trace the connection not just in biochemistry, but in evolution, in ecology, in the ancient DNA of organisms that have been exchanging chemistry since before there were flowers. You ate something today that grew in the ground, something that has been finding its way to human bodies for longer than human civilization, longer than language, longer than the first tools. And right now, in the quiet of wherever you are, the chemistry of that thing is doing its work. The same work it has always done. Not because it knows you need it, not because the plant intended it, but because the fit is so old, so worn in, so deepened by time, that it simply happens now the way breathing happens, without thought, without effort, without anyone on either side of the exchange needing to understand it. The potassium from that banana, if you ate one, is already in your bloodstream. The lycopene from whatever tomato crossed your path today has already been absorbed into tissues that needed it. The quercetin from an apple, if there was an apple, has already met the gut bacteria that will transform it into something your cells can use directly. All of this running quietly in the background. Like the soil running quietly in the background. Like the fungal networks threading through the dark beneath every patch of ground you have ever walked on, carrying things from one root to another, maintaining a chemistry that has been uninterrupted for hundreds of millions of years. I want to spend some time with the mycorrhizal network because I mentioned it in passing earlier, and it deserves more than a passing mention. This is one of those things that, when you understand what it actually is, what it actually does, changes the way you look at every forest, every field, every patch of ground. The word mycorrhiza means roughly fungus root. Myco for fungus, rhiza for root. The mycorrhizal relationship is the partnership between fungi and plant roots, and it is not a rare or exotic thing. It is the default state of most plants on Earth. Something between 80 and 90% of all land plants form mycorrhizal associations. Not all plants, there are exceptions, but most plants, most of what grows. The fungi involved are not the mushroom caps you see above ground, though those are the reproductive structures of some mycorrhizal species. The organism itself is the underground network of hyphae, which are filaments thinner than a human hair, extending through the soil in densities that are almost impossible to visualize. In a single teaspoon of healthy forest soil, there can be kilometers of fungal hyphae. Not meters, kilometers. These hyphae form networks. They connect to plant roots. They extend outward from those roots through soil pores too small for roots to enter. And they connect to other plants. A single fungal network can, in some cases, connect dozens of trees across hectares of forest. The first serious scientific attention to this phenomenon came from a researcher named Suzanne Simard, who was working in Canadian forests in the 1990s. She was trying to understand why trees in natural forests seemed to survive better than trees in managed plantations, even when the plantations had better soil and more sunlight and fewer competing plants. She designed an experiment involving radioactive carbon. She labeled carbon dioxide so that it could be traced, and she allowed a birch tree to absorb it through photosynthesis, turning it into sugar. She then tracked where that carbon went. It went underground, through the roots, into the fungal network, and from the fungal network into the roots of a fir tree several meters away. The carbon the birch tree had made from sunlight was being shared with the fir tree through the fungal network. Not all of it, a fraction, but a measurable, traceable fraction, moving from one organism to another through an underground connection. Simard went on to show that the transfer was not random. It followed patterns. Trees in shade, receiving less sunlight, tended to receive more carbon from the network than they gave. Trees in full sun, producing more sugar than they needed, tended to give more to the network. The network behaved in some ways like a resource distribution system, like a commons. Later research has added detail and nuance. Not every transfer is altruistic in the biological sense. Fungi have their own interests. The dynamics are complex. But the basic fact that carbon, nutrients, and even chemical signals move between plants through fungal networks is well established. And among the signals that move through these networks are chemical warnings. When a plant is attacked by insects, it releases chemical signals from its roots. These signals travel through the mycorrhizal network to neighboring plants. The neighboring plants, receiving the signal, begin to produce their own defensive compounds before they themselves are attacked. A warning system, underground, moving at the speed of chemistry rather than the speed of nerve impulses, which is to say slowly, but it moves. The forest is not a collection of individuals competing for resources. It is a community with a shared nervous system of sorts, a distributed awareness, not consciousness, not intention, but information moving between organisms in ways that change their behavior. And this community, this living, communicating ground, is what produces the plants that eventually produce the food that eventually finds its way into the human body. The walnut on the tree in October, the tomato on the vine in August, the blueberries in the field in July. They are all the products not just of their own genetics, not just of rain and sunlight, but of the mycorrhizal networks threading through the soil beneath them, the bacterial communities in the rhizosphere around their roots, the whole invisible community of the ground that shapes the chemistry the plant makes. When you eat something that grew in the ground, you are not just eating the plant. You are, in some extended and attenuated sense, eating the community that made the plant possible. Let's come back to the doctrine of signatures. I mentioned it earlier, and I want to be careful here, because it is easy to either overstate its importance or dismiss it too quickly. both would miss something real. The doctrine of signatures is a very old idea. Its clearest articulation came from a Swiss physician named Paracelsus in the 16th century, and later from a botanist named William Coles in the 17th. The idea was that God, or nature, had marked plants with visible signs indicating their medicinal uses. A plant that looked like a body part would heal that body part. A yellow plant would heal jaundice. A plant with red sap would be good for blood disorders. Most of this is wrong, substantially wrong. The correspondence between visual appearance and medicinal use is not reliable enough to be used as a diagnostic system. Treating it as a reliable guide to medicine would be dangerous. But here is the part that keeps me thinking about it. In a handful of cases, the correspondence is real. And real not just coincidentally, but for mechanistic reasons. The walnut. The resemblance to a brain is visual, structural. The medicinal benefit to the brain is real, chemical. The connection between appearance and function is not causal, but the appearance and the function coexist in the same object. The plant makes the shape and makes the compound. Both are real. Their connection is evolutionary, not cosmological. The kidney bean. Kidney-shaped, as the name suggests, and kidney beans do contain compounds, particularly flavonoids and saponins, that have been shown to have effects on kidney function, on blood pressure regulation, on the filtration processes the kidney performs. The correspondence is imperfect, but it is not nothing. The walnut of the eye. Slice a carrot crosswise and you will see a pattern of radial lines around a central pupil that genuinely resembles an eye. Carrots contain beta-carotene, which the body converts to vitamin A, which is essential for the maintenance of the photoreceptor cells in the eye. A deficiency of vitamin A causes night blindness and eventually full blindness. The compound in the carrot that makes the eye is the compound the eye needs. Now, none of this proves the doctrine of signatures. The carrot does not look like an eye because God designed it to indicate its purpose. The carrot looks like an eye because of the geometry of how carrot tissue is organized. The carrot contains beta-carotene because of the pigmentation chemistry of that particular plant lineage. The eye needs beta-carotene because of the specific biochemistry of vision. These three things happen to align, but the alignment is not the point. What is interesting about the doctrine of signatures, viewed from this angle, is what it says about human observation. Humans have been noticing these connections for a very long time. Not chemically, not with instruments, with eyes. With the kind of pattern recognition that is essentially the same faculty that lets us find faces in clouds and animals in star patterns. A faculty prone to false positives, but also, occasionally, to true positives. The people who developed the doctrine of signatures were looking at the world and noticing correspondences. Most of the correspondences they found were wrong, but some were right, not for the reasons they thought, not through divine intention, but through the real evolutionary relationships between plants and the bodies that eat them. The human pattern recognition system, turned toward the natural world, occasionally stumbled on real relationships, found them accidentally, encoded them in folk medicine, passed them on. And the science, centuries later, confirmed the pattern even while correcting the explanation, which is, in itself, a kind of co-evolution. Between the human mind and the natural world, between observation and what there is to observe. I said I wanted to talk about the specific way certain phytonutrients interact with human cell receptors. There is one story in particular I keep returning to: resveratrol. Resveratrol is a polyphenol made by grapes and by several other plants as a defense against fungal infection. When a grapevine is under fungal attack, it upregulates its production of resveratrol. The compound inhibits fungal growth. This is a well-characterized defense response. In the human body, resveratrol activates a family of proteins called sirtuins. Sirtuins are involved in one of the most fundamental processes in biology, the regulation of gene expression in response to stress and caloric restriction. When an organism is experiencing caloric restriction, getting less food than it would prefer, sirtuins help orchestrate a shift in cellular behavior toward repair and conservation. DNA repair rates increase. Autophagy, the process by which cells break down and recycle damaged components, increases. Inflammatory responses are modulated. This suite of responses is associated, in animal studies and some human studies, with extended healthy lifespan. Organisms under moderate caloric restriction consistently show markers of better cellular maintenance, reduced age-related damage, extended period of healthy function before decline. Resveratrol appears to partially mimic this response. Not because it evolved to do so, not because the grapevine was making resveratrol to extend the lifespans of the animals that ate it. The grapevine was making resveratrol to kill fungi. But in the human body, the sirtuin pathway happens to be activated by this molecule. The human sirtuin proteins happen to respond to resveratrol with the same changes they make in response to caloric stress. This is one of those places where the accidental chemistry becomes very hard to characterize simply. The plant was solving one problem. The human biology was solving another problem. The solution to one problem fits the mechanism of the other with enough precision that the same molecule activates both. The research on resveratrol in humans is mixed and complicated, much of it done with doses far higher than you would get from eating grapes, and the bioavailability questions are significant. But the basic fact of sirtuin activation by a plant compound is real, and what it hints at is something larger. That the overlap between plant chemistry and human biology is not limited to nutrition, not limited to the provision of vitamins and minerals and basic nutrients. It extends into signaling, into the regulation of gene expression, into the chemical language by which cells communicate with each other and adjust their behavior in response to conditions. Plants have been speaking a chemical language for hundreds of millions of years, and some of what they are saying, it turns out, human cells can hear. There is a compound in broccoli and other brassicas, Brussels sprouts, cabbage, cauliflower, kale, called sulforaphane. It is formed, like allicin in garlic, by enzymatic reaction when the plant is damaged. A precursor compound called glucoraphanin reacts with an enzyme called myrosinase when cells are broken, producing sulforaphane. The plant makes it as a pest deterrent. The same two-stage delivery system as garlic. The compound activated by damage. The chemistry turned on specifically at the moment of attack. In the human body, sulforaphane activates a transcription factor called NRF2, which is a master regulator of the body's own antioxidant defenses. NRF2 activation turns on a suite of genes that produce antioxidant enzymes. Enzymes that neutralize free radicals and reduce oxidative stress at a rate far beyond what any dietary antioxidant can do directly. This is a distinction worth staying with. Most dietary antioxidants work by directly neutralizing free radicals. They absorb the damage. They are consumed in the process. You need a constant supply of them. Sulforaphane does something different. It does not directly neutralize free radicals. It turns on the machinery that does. It activates the body's own antioxidant production system. The effect is therefore amplified and sustained in a way that direct antioxidants cannot match. A compound that plant made to deter caterpillars activates the most powerful antioxidant defense system the human body possesses. The caterpillar and the human responding to the same chemistry in completely different ways. The plant, in the middle, making the compound for reasons that have nothing to do with either of them specifically. And yet the fit. This is something that came up in a recent episode of Sleep Science, or at least it came up in conversations around the research for it, and it has stayed with me. The idea that the human body's relationship with plants is not primarily about getting things from plants that the body cannot make. Sometimes it is. Vitamin C is an example of that. Folate from food is an example of that. But in many cases, the phytonutrients are not replacing something the body cannot make. They are stimulating or modulating systems the body already has. They are not filling gaps. They are turning knobs. The sulforaphane turning up NRF2, the resveratrol activating sirtuins, the curcumin inhibiting NF-kappa B, the anthocyanins modulating the MAPK signaling pathways involved in inflammation and cell survival. These are not the body using plant chemistry as a substitute for its own chemistry. These are the body's regulatory systems responding to plant chemistry as inputs, interpreting plant compounds as signals, adjusting accordingly. Which means the human body, in some sense, evolved not just to tolerate phytonutrients, but to be regulated by them, to use them as information, to incorporate the chemical output of plants into the ongoing self-regulation of its own systems. The plants are not just feeding us. They are, in some extended sense, in conversation with us, sending signals that our cells have learned across evolutionary time to listen to. And when those signals are absent, the regulatory systems lose some of their inputs. They are calibrating against nothing. They are running without the information they evolved to receive. This is not the only reason for chronic disease. It is not a simple story. But it is a real thread in the story of why the diet of the industrial age, depleted of plant diversity and phytonutrient density, is associated with health outcomes different from the diets that preceded it. Not just what has been added, what has been removed. What signals are no longer arriving? What conversations have gone quiet? Let's talk about what the soil is doing differently. There is a mineral called selenium. It is an essential trace element for the human body. Essential, meaning necessary in small amounts, toxic in large amounts, and deficient in many populations. Selenium is incorporated into proteins called selenoproteins, of which the human body makes about 25 distinct varieties. These selenoproteins include glutathione peroxidase, one of the body's primary antioxidant enzymes. They include thyroid hormone-converting enzymes. They include proteins involved in immune function and DNA synthesis. The body cannot make selenium. It must come from the diet. And it comes primarily from plants and animals that have accumulated it from the soil. The concentration of selenium in plant food depends almost entirely on the concentration of selenium in the soil where the plant grew. Selenium is not evenly distributed in the Earth's crust. Some soils are selenium-rich. Some are selenium-poor. In selenium-poor regions, populations that eat locally grown food consistently show signs of selenium deficiency. The selenium in the soil moves through the roots into the plant, into the seeds or leaves or fruit, into the animal that eats the plant or the human that eats it directly. The trace elements of the Earth's crust, distributed unevenly across the planet's surface, find their way into the chemistry of life through this chain. The soil is the bridge. And it is not just selenium. It is iodine, critical for thyroid function. It is zinc, essential for hundreds of enzyme systems. It is magnesium, involved in over 300 biochemical reactions. It is chromium, iron, manganese, molybdenum, each one needed in specific amounts, each one entering the body primarily through what grew in the ground. The diversity of the soil is, ultimately, the diversity of the body's chemistry. A depleted soil, over-farmed, over-fertilized with only the three macronutrients, nitrogen, phosphorus, potassium, depleted of trace minerals, produces plants that are nutritionally simpler than what grew from richer soil. The plants grow. They look normal. They have the right calories. But the trace element content is lower. The phytonutrient density is often lower. The chemistry that the living soil community produces and makes available to the plant's roots is absent. Several large analyses comparing the nutrient content of vegetables and fruits from the 1950s to measurements made in the 1990s and 2000s found significant declines in mineral content. The vegetables were not less vegetable-shaped, but they were less minerally rich. The soil had changed. The plants reflected the soil. The conversation between ground and body had been interrupted at the source. Not dramatically, not catastrophically, subtly, gradually, in ways that are difficult to measure against a baseline that was itself always changing. But the direction was clear. I want to slow down here because we have covered a lot of ground, and I do not want any of it to feel like a list. Think about what has actually been described. A plant makes a compound to protect itself from fungi, and that compound activates the human body's master antioxidant switch. A plant makes a pigment to attract birds, and that pigment reduces cardiovascular disease in humans. A plant makes a chemical to deter biting insects, and that chemical turns out to be one of the most powerful antimicrobial agents in the natural world. A plant makes a root pigment to survive the microbial world of the soil, and that pigment reduces chronic inflammation in the human body. The soil bacteria make a molecule as part of their own metabolism, and when that molecule reaches the human brain, it increases serotonin production. The fungi threading through the soil produce nutrients that trees could not otherwise access, and those trees produce the fruit that contains the phytonutrients that regulate human cell signaling. Each one of these connections has a clear, clean evolutionary explanation. Each one makes perfect sense as a product of coevolution, of the long, wordless negotiation between chemistry and survival across geological time. But standing back and looking at all of them together, at the sheer number of them, at how deep and specific each connection is, at how many systems in the human body seem to have been shaped specifically in response to the chemistry of the ground, there is a feeling that is hard to dismiss. Not a mystical feeling, not a religious one, not a feeling that requires the ground to be conscious or the earth to have plans, just a feeling of scale. A feeling that the relationship between what grows from the ground and what lives above it is so old and so deep and so thoroughly woven into the biology of both, that the distinction between food and medicine, between nutrition and regulation, between the ground and the body, starts to feel somewhat arbitrary. We are not organisms that happen to eat plants. We are organisms that were shaped, continuously, by plants, for whom the chemistry of the plant world is not external input, but internal calibration, for whom the soil microbiome is not just background, but part of the system, for whom the absence of the chemical conversation that the ground has always been having with the body is a genuine disruption of something very old and very deep. I want to come back to something that was briefly mentioned earlier and has been waiting for more space. The gut microbiome. The human gut contains somewhere between 38 and 100 trillion microbial cells. The estimates vary depending on who is counting and how. But even the conservative number is extraordinary. More microbial cells in the gut than there are human cells in the entire body. These organisms are not passengers. They are participants. They digest fibers that human enzymes cannot touch, producing short-chain fatty acids that feed the cells lining the gut. They produce vitamins. They train the immune system. They influence the production of neurotransmitters. They regulate inflammation. And they are fed, primarily, by what we eat. The diversity and composition of the gut microbiome changes in response to diet. A diet rich in plant diversity, in different vegetables and fruits and legumes and whole grains, with the wide range of fibers and polyphenols and other plant compounds that diversity provides, produces a more diverse and resilient gut microbiome. A diet poor in plant diversity produces a simpler microbiome. Fewer species, less functional redundancy, less ability to perform the full range of tasks the system evolved to perform. And this matters because the gut microbiome is not a separate system. It is continuous with the immune system, with the nervous system through the gut-brain axis, with metabolic regulation through the compounds it produces and the signals it sends. The plant world, through the gut microbiome, has a reach that extends far beyond nutrition in the simple sense. The phytonutrients and fibers from the plant world feed and shape the microbial community that feeds and shapes the human body's most fundamental regulatory systems. There is a loop here, a circular conversation. The soil microbiome shapes the plant chemistry. The plant chemistry feeds the gut microbiome. The gut microbiome shapes the human body's regulation of everything from inflammation to mood. And the human body, through what it excretes and what it plants and how it manages the land, shapes the soil. The loop has been running for millions of years. The participants at each stage have been selected for by the loop itself, each one shaped by the others, all of them, in some sense, part of a single extended system that has never quite had a name, because the name would have to span soil science and nutrition science and immunology and neuroscience and ecology and evolutionary biology all at once. Polyphenols are transformed by gut bacteria. This is a detail that seems small and turns out to be important. When you eat a blueberry, the anthocyanins in it are not immediately absorbed in the stomach. They pass into the intestine, where specific gut bacteria, particular species that have coevolved with polyphenol-consuming primates, metabolize them, break them down, transform them into smaller molecules called urolithins and other secondary metabolites. These transformed molecules are sometimes more bioavailable than the original compound, sometimes more biologically active. The gut microbiome is not just feeding on polyphenols. It is transforming them into the forms that the human body can use most effectively. The relationship is three-way: plant, gut microbe, human body, each one shaped by the others. The plant making the polyphenol for its own reasons. The gut bacteria evolved to process it. The human body evolved to absorb and use what the bacteria produce. Remove any one of the three, and the system works less well. A diet with no polyphenols starves the gut bacteria that process them. Those bacteria decline in the population. The transformation is less efficient. The human body gets less of what the plant was providing. This is part of why the specific matrix of food, the whole food, rather than the isolated compound, tends to work better than supplementation. The polyphenol in a capsule bypasses the gut bacteria that transform it. The fiber in the whole berry feeds those bacteria and sustains the microbial community that makes the transformation possible. The whole system functions best as a whole system, which is not an argument against supplements in specific situations of deficiency, but it is an observation about how the system evolved. It evolved as a three-way relationship. The supplement disrupts one of the relationships to deliver a simplified version of one component. Sometimes that is necessary. Sometimes it is not the same as the whole. There is a specific type of fiber called inulin that I find quietly remarkable. Inulin is found in chicory, Jerusalem artichokes, garlic, onions, asparagus, leeks, bananas, and several other plants. It is not digestible by human enzymes. It passes through the stomach and small intestine unchanged and arrives in the large intestine, where specific gut bacteria ferment it. These bacteria are called Bifidobacterium and Lactobacillus species, and they are central members of the healthy gut microbiome. They feed on inulin. When they ferment it, they produce short-chain fatty acids, primarily butyrate, propionate, and acetate. Butyrate is the primary fuel source for the cells lining the colon, the colonocytes. They use it almost exclusively, preferring it to glucose. Without butyrate, the colon cells begin to function less well. The barrier between the gut contents and the bloodstream becomes more permeable. Things that should stay in the gut begin to leak across into the blood, triggering immune responses and chronic low-grade inflammation. Inulin is a fiber that humans cannot digest, which feeds bacteria that produce a compound that feeds the cells that maintain the integrity of the gut barrier. Without the fiber, the bacteria are less productive. Without the bacteria, the butyrate is not produced. Without the butyrate, the gut cells are less healthy. Without the healthy gut cells, the barrier is compromised. The human body, in other words, depends on bacteria to extract from plants something that human enzymes cannot extract directly. The plant provides the substrate. The bacteria do the work. The human body receives the product. This is not an optional add-on to the digestive system. It is the digestive system, a substantial part of it, involving organisms whose relationship with the human gut goes back millions of years, and whose role in human health is far more fundamental than the word microbiome sometimes implies. There is a carrot grown in the mountains of Afghanistan that looks nothing like the orange carrots in a supermarket. It is purple, or yellow, or white. The orange carrot is actually a relatively recent development. It was bred in the Netherlands in the 17th century, possibly selected for its colour to honour the Dutch House of Orange, though the horticultural historians debate this. The original wild carrot was white. The colour variations that developed in Central Asia, where the carrot was first domesticated around 1,000 years ago, were purple and yellow, from anthocyanins and carotenoids respectively. The beta-carotene that gives modern carrots their orange colour and their benefit to eye health is present in much smaller amounts in the white ancestor of the carrot. It was concentrated by selective breeding, which raises an interesting question about the whole conversation between the ground and the body. The conversation is not static. It did not end when humans appeared. It has continued, and humans have been participants in it in an active way, not just receiving what the ground offers, but changing what grows, shaping plant chemistry through selection and breeding, and eventually genetic modification, changing the terms of the conversation. Agriculture is, in a sense, the most intensive period of human participation in the co-evolution between plants and the human body. We did not passively receive what grew, we changed what grew. We made wheat into something very different from wild grass. We made corn into something unrecognizable from its ancestor, teosinte. We made 1,000 varieties of apple from a tree that grew wild in Kazakhstan. Some of these changes improved the nutritional density of the food. Some reduced it, selecting for sweetness or yield at the expense of phytonutrient complexity. Some changed the chemistry in ways that are only now being understood. But the basic direction of the relationship, the direction that ran for hundreds of millions of years before the first human deliberately planted a seed, was coevolutionary. The ground and the body shaping each other across time. And that deep direction did not change just because humans became active participants in it. It continued. We became part of the evolutionary pressure on plants, and plants continued to be part of the evolutionary pressure on us. There is a study, or there are several studies really, suggesting that populations that have been growing and eating a particular food for many generations show specific genetic adaptations that populations without that history do not. Lactase persistence, the ability to digest milk sugar into adulthood, is the most famous example. But there are others. Amylase copies, duplications of the gene that makes the enzyme that digests starch, are more numerous in populations with long histories of eating starchy foods. Certain polymorphisms in fatty acid metabolism genes are more common in populations with long seafood eating traditions. The food changed the humans who ate it. Over generations. Gradually, invisibly, through the same process that changed every other part of the relationship. The fit deepened. The humans most adapted to the available food survived and reproduced more. The food, shaped by humans, changed in response to what humans selected for. The conversation continued, just with new participants in a more active role. I want to come back to berries one more time. There is a specific mechanism I glossed over earlier that deserves more attention. The anthocyanins in dark berries do not just reduce oxidative stress in a general sense. In the brain specifically, they appear to improve the efficiency of a process called neuroplasticity. The ability of the brain to form new connections between neurons, to strengthen existing connections, to change its own structure in response to learning and experience. This process depends on a protein called BDNF, brain-derived neurotrophic factor. BDNF is sometimes described as fertilizer for the brain. It promotes the survival of existing neurons. It stimulates the growth of new ones in brain regions where neurogenesis continues into adulthood, particularly the hippocampus, which is centrally involved in memory and spatial navigation. It strengthens synaptic connections. Anthocyanins increase BDNF expression in the brain. They do this by inhibiting certain enzymes that break down the signaling molecules that activate BDNF production, and by modulating the expression of the gene itself through pathways involving the transcription factors the brain uses to regulate its own plasticity. A compound made by a berry plant to protect its outer cells from UV radiation ends up in the human brain, promoting the growth of new neurons and the strengthening of memory circuits. These two things, UV protection in a berry and neuroplasticity in a mammalian brain, are not obviously related. The chemistry that connects them is complicated and indirect, but the connection exists, has been demonstrated in careful studies, is not in serious scientific doubt. And what the connection suggests, once again, is that the plant and the brain have been exchanging chemistry for long enough that the plant's solutions have become the brain's inputs. Long enough that the brain's plasticity is partially calibrated to the presence of these compounds. Long enough that their absence, in a diet lacking in dark berries and other flavonoid-rich foods, may have real effects on the brain's ability to maintain and build itself. Not dramatic effects, not effects that would manifest in a week, but effects that accumulate over a lifetime. Small reductions in the inputs the brain's self-maintenance system evolved to receive. Small decrements in neuroplasticity and neuroprotection, compounding over decades. Which is not a reason for alarm. It is just a reason for quiet, sustained appreciation of the blackberry, of the blueberry, of the purple carrot, should you find one. Of the fact that what grows in the ground and what keeps the brain working have been in conversation for so long that they are, in some chemical sense, part of the same conversation. The vitamin D story is interesting here because it is almost the inverse of all the others. Vitamin D is not a plant nutrient. You do not get much of it from plant food. You get most of it from sunlight, specifically from UVB radiation triggering its synthesis in the skin from a cholesterol precursor. But the mushroom is an exception. Mushrooms, when exposed to sunlight, synthesize vitamin D2 in their outer tissue, from the same precursor chemistry that occurs in human skin. Fungi and humans, both making vitamin D from sunlight, through a biochemical pathway so similar that it almost certainly represents a shared ancient inheritance, a process that evolved in a common ancestor before the split between fungi and animals hundreds of millions of years ago. Vitamin D receptors are found in almost every tissue in the human body. The compound acts more like a hormone than a vitamin, regulating the expression of hundreds of genes involved in immune function, cell division, inflammation, calcium metabolism, and brain function. And the pathway that makes it, the activation of a precursor by UV light, exists in mushrooms. The ground, or at least what grows from it, including the fruiting bodies of fungi, can provide something that most plant food cannot. Leave mushrooms on a sunny windowsill for a few hours, and their vitamin D content rises dramatically. They are, in effect, doing the same thing human skin does in sunlight, using the same ancient chemistry, preserved across the hundreds of millions of years since the last common ancestor of fungi and animals, for the same ultimate purpose. Which is another version of the same theme. The ground and the body sharing chemistry that predates both of them in their current form. Chemistry inherited from a shared past, maintained in parallel in organisms that look nothing like each other and share almost no visible features. The connection between a mushroom and a human being is not obvious from the outside. But inside, at the molecular level, there is a process running in both of them that is essentially the same process, refined slightly differently by a few 100 million years of independent evolution, but recognizably the same. There is a compound in green tea called L-theanine. It is an amino acid. It does not appear to be present in significant amounts in any other widely consumed food. It is made by the tea plant, Camellia sinensis, and it is found primarily in the leaves. What it does in the human body is unusual. L-theanine crosses the blood-brain barrier. In the brain, it promotes alpha wave production. Alpha waves are a specific pattern of electrical activity associated with a relaxed but alert state. The state you might be in during meditation, or when you are doing something you find engaging but not stressful. L-theanine does not cause sedation. It does not impair cognitive function. It appears to promote a kind of calm clarity. It also works synergistically with caffeine, which is also in tea. The caffeine provides alertness and focus. The L-theanine smooths the edge of the caffeine, reducing the anxiety and jitteriness that caffeine alone sometimes produces. The two compounds together produce a state that many people describe as different from the effect of either one alone. More settled, more focused. The tea plant makes L-theanine as part of its nitrogen metabolism, as a way of storing and transporting nitrogen through the plant. It is not made as a defense or an advertisement. It is just a byproduct of the plant's internal chemistry. And in the human brain, it does something specific and measurable and, depending on your circumstances, quite useful. That is the pattern, isn't it? The plant making something for its own purposes. The human biology having a response to it that is specific enough to be surprising. Not a general response to a general compound, a precise, mechanism-specific interaction with a molecule the plant made for completely different reasons. The brain has alpha-wave promoting responses to an amino acid that a camellia plant evolved for nitrogen storage. The fit between the molecule and the receptor, between the plant chemistry and the neural chemistry, is not approximate. It is precise. And the precision did not arise because the tea plant was trying to calm human brains. It arose because human brains evolved in an environment where this compound was present, and the individuals whose brains responded to it in functional ways, who could find calm focus when the compound was available, did slightly better, survived more reliably, passed the trait on, quietly, the way all evolutionary advantages pass on, without fanfare, without anyone noticing, just one generation slightly more fitted to the world than the one before it. And the world, in this case, included the tea plant. The tea plant that was growing in the forests of southern China and Southeast Asia for millions of years before anyone thought to pick its leaves and steep them in hot water. The plant just growing, making L-theanine for nitrogen. Not knowing, in any sense that involves knowing, that something in the vicinity was developing a brain that would one day need exactly what it was making. Right now, tonight, while you are lying in the dark, the ground beneath you is not resting. It never rests. The soil is metabolizing. Billions of organisms running their chemistry in the dark, converting one thing to another, cycling nutrients through forms, breaking down what was alive and building what will be alive again. The bacteria are oxidizing minerals. The fungi are threading new hyphae through pores in the earth, looking for something, finding something, always extending the network by another fraction of a millimeter. The roots are growing. Roots grow at night. During the day, a plant puts its energy primarily into photosynthesis, into the chemistry of the leaves and the fruit. At night, with no light to drive photosynthesis, the plant redirects its resources downward. The roots elongate. They branch. They probe. They find new territory in the dark. The root tip is one of the most sensitive structures in the plant world. It carries a small cluster of cells that function, in a loose sense, as a navigation system. These cells detect gravity, which tells the root which way is down. They detect moisture gradients, which tell the root where water is. They detect chemical signals from the soil microbiome, which tell the root where nutrients are concentrated and where other roots are competing. The root tip is exploring, tasting the soil, following gradients, finding what it needs. And as it grows, it releases compounds from its tip: sugars, amino acids, organic acids, specific signaling molecules. These attract particular bacteria and fungi. The root is not just finding the soil community, it is selecting it, recruiting the microbes it wants, building the rhizosphere community that will serve it best in this particular place, in this particular season, under these particular conditions. The plant, in other words, is not passive in the soil. It is active. It is communicating. It is managing the community around it with a chemical sophistication that we are only beginning to map. And all of this is happening right now, in the dark. Beneath every field and forest and garden and crack in the pavement where a weed has found enough soil to take root. The ground is working. The ground is always working. And some of what it is producing tonight, some root being grown, some mineral being made available, some compound being synthesized in response to the soil's chemical signals, will eventually make its way into something that will be eaten. Something that will cross from the outside world into the inside of a body and find its way to a cell that has been waiting for it, in the evolutionary sense, for millions of years. There is a specific example I want to spend time with, because it illustrates something about the precision of these connections that I have not quite captured yet. Folate, which I mentioned earlier, is made by plants as part of their one-carbon metabolism. The same one-carbon chemistry that plants use to copy their DNA is essential for humans copying their DNA. We inherited this chemistry from the same ancient ancestors. But there is a gene, or a variant of a gene, that a significant proportion of humans carry, called MTHFR. The letters stand for methylene tetrahydrofolate reductase, which is the enzyme the gene codes for, the enzyme that converts folate into its active form in the body. People with certain variants of this gene convert folate less efficiently. The enzyme works more slowly. The active form of folate accumulates less quickly, which means, in practical terms, that these individuals need more dietary folate to maintain the same cellular function as someone without the variant. The MTHFR variants are not rare. Depending on which variant and which population, somewhere between 10 and 40% of people carry one of the more significant forms, which means a substantial fraction of the human population has a genetic need for higher folate intake that the average person does not. And the foods highest in folate, the dark leafy greens, the legumes, asparagus, the avocado, these are foods that have been central to human diets in the regions where these variants are common for thousands of years. Not because anyone knew about MTHFR, not because ancient farmers were selecting diets based on genetic variants, just because these foods grew where people lived, because people ate what grew, because, over enough generations, the genetic variants that required these foods were maintained in populations where these foods were available, because those populations did not suffer meaningfully from the deficiency that the variant creates when food is adequate. It is a loop that runs in the other direction. Not just the body adapting to the plant's presence over generations, shaping which genetic variants persist in the population. The food and the genome in conversation, each one influencing the other across time. The fit between what grows in the ground and what the human body needs is not just about the average human body. It is, in some cases, about specific variants of specific genes in specific populations, matched with specific plants that grew in those populations' historical environments. The specificity goes all the way down. Let me come back to something that might seem small, but has been sitting in the background of everything I have said tonight. The way food is eaten. Not just what is eaten, how. There is research suggesting that the mechanical process of chewing plants, the act of breaking cell walls, of mixing plant tissue with saliva, itself affects the chemistry of what is absorbed. Chewing releases enzymes from the plant. It mixes plant enzymes with salivary enzymes. It begins chemical reactions that continue as the food moves through the digestive system. Broccoli, for instance. If you cut broccoli and then cook it immediately, the myrosinase enzyme that converts glucoraphanin to sulforaphane is deactivated by the heat before it can do much work. But if you cut it and wait several minutes before cooking, the conversion happens at room temperature while the enzyme is still active. And then, the cooking locks in the sulforaphane that has already been made. The sulforaphane itself is heat stable. The enzyme is not. This is not obscure or esoteric information. It is simple biology. But it suggests that the way a plant is prepared, the specific sequence of cutting and resting and cooking, affects the chemistry that the human body ultimately receives. Traditional food preparation often, not always, but often, happens to maximize these conversions. Fermentation, long cooking with acid, combining specific spice and vegetable combinations like turmeric and pepper, cutting and resting before cooking. The practices that persisted were practices that worked, in the sense of producing food that sustained and benefited the people who ate it. The ones that didn't work stopped being practiced. Another form of coevolution. between human culinary practice and plant chemistry, between what people do with food and what the food does in the body. The tradition carrying the knowledge that the science later confirmed. There is something I want to say about bitterness. Bitter is, of all the tastes, the one with the most complex relationship to human health. Sweet is primarily a signal of calories, of energy, of the ripe fruit that was safe and valuable and worth eating. The evolutionary argument for the sweetness preference is straightforward. Bitter is different. Bitter evolved as a warning signal. Many toxic compounds, particularly plant alkaloids that plants produce as defenses against herbivores, are bitter. The bitterness is the plant's signal that it does not want to be eaten by the wrong animal. Humans are exceptionally sensitive to bitter compounds. We can detect them at concentrations far lower than we can detect sweet or salty or sour. This hypersensitivity makes sense as a survival trait. A small amount of a toxic alkaloid, detected before the animal has consumed enough to harm itself. But here is the thing. Some of the most beneficial plant compounds are also bitter. Polyphenols are often bitter. Many of the flavonoids that have the most significant health benefits are perceived as bitter. The compounds in dark chocolate, in coffee, in cruciferous vegetables, in many herbs and spices, these are often bitter compounds that the plant made as defenses and that the human body, in moderate quantities, finds beneficial. The bitterness, in these cases, is real. The plant really is saying, in its chemical way, do not eat too much of this. The alkaloids really are present. The plant really did evolve bitterness as a deterrent. But the human relationship with bitterness has been shaped by something more complicated than simple avoidance. Humans learned, over generations, to tolerate and even appreciate bitterness in certain contexts. To associate bitter foods with the benefits they provided. To develop a taste for compounds that were initially aversive because the long-term benefit outweighed the short-term unpleasantness. This tolerance for beneficial bitterness is itself an evolved trait. The humans who could eat bitter plants and gain their benefits, who did not reject the broccoli and the dark leafy greens entirely because of their bitterness, had better access to the phytonutrients those plants provided. And the plants, in turn, were not uniformly bitter. They modulated their chemistry. The outer leaves of a plant, most at risk from grazing, might be more bitter. The fruit, intended to be eaten, might be sweet. The timing of bitterness changed with season. The concentration varied with stress. The animals in those environments were selected for increasingly nuanced responses to bitterness, not simple avoidance. A more complex relationship, in which bitterness was a warning that sometimes meant stop and sometimes meant proceed carefully. That is the relationship humans have with bitterness now. Complicated, context-dependent, acquired over time. A relationship that was shaped by the plants and by the compounds those plants made for their own protection, and by the bodies of the animals that ate those plants and were slowly, over generations, changed by what they ate. I want to come back one more time to the soil. There is a specific microorganism I have not mentioned yet that I think deserves a moment. Azotobacter is a genus of soil bacteria that does something that seems, when you really look at it, almost magical. It takes nitrogen from the air. The air is 78% nitrogen. Nitrogen is one of the primary limiting nutrients for plant growth, and therefore for essentially all terrestrial life. Without nitrogen, proteins cannot be made, DNA cannot be made, life cannot happen. But the nitrogen in the air is molecular nitrogen, two nitrogen atoms bonded to each other so tightly that very little chemistry can break the bond. Plants cannot use it. Animals cannot use it. The nitrogen is there, everywhere, in every breath, completely unavailable. Azotobacter breaks the bond. It has an enzyme called nitrogenase that splits molecular nitrogen and converts it into ammonia, which plants can absorb through their roots. This process, nitrogen fixation, is one of the most energetically expensive reactions in biology. Azotobacter spends an extraordinary proportion of its metabolic budget on it. Why does it do this? Not for the plants, not for the ecosystem. Azotobacter makes ammonia for its own nitrogen needs, and some of that ammonia leaks out into the surrounding soil and becomes available to plants. But the result is that a bacterium in the soil, working for its own metabolic purposes, accidentally makes the most limiting nutrient in terrestrial ecosystems available to the rest of life. Without nitrogen-fixing bacteria like Azotobacter, and its more famous relatives like Rhizobium, which live in root nodules of legumes and fix nitrogen in even greater quantities. Without these organisms quietly working in the soil, running their expensive chemistry for their own reasons, terrestrial plant life as we know it would be impossible. The entire food web, every plant, every animal that eats a plant, every human who has ever lived, all of it ultimately dependent on bacteria in the soil doing their chemistry. The ground, working in the background, always working, making the life of the surface possible, not knowing that it is doing so, not needing to know, just chemistry. Patient, ancient, running. Let's come very gently to the question that underlies everything that has been said tonight. What does it mean that the ground grows exactly what the body needs? Not in every case, not perfectly, not without variation and deficiency and the occasional mismatch. The fit is not perfect. Evolution is not an engineer. It is a process, messy and incremental, that produces approximate solutions to changing problems. But in the broad pattern, across the millions of specific connections between plant chemistry and human biology, across the co-evolutionary history of animals and the plants they ate, across the ancient shared biochemistry that goes back further than animals or plants as categories, there is something real. The ground and the body have been in conversation for so long that the line between them is not quite where we think it is. You are made of what grew in the ground, not just metaphorically, literally. The carbon in your body was fixed from the air by plants. The nitrogen was fixed from the air by bacteria. The minerals in your bones came from the soil, through the roots of plants, through the food chain to you. The water in your cells cycled through the ground and through plant roots and through the atmosphere before it was yours. There is no part of you that did not come from somewhere else. And the somewhere else is ultimately the ground. And the ground continues to shape you, not just through the macronutrients, through the precise chemistry of phytonutrients and trace elements and microbial signals. The plants you ate yesterday are still working. Their chemistry is still in conversation with your cells. The signals are still being sent and received. The last meal you ate today, whatever it was, is part of a conversation that started before the human species existed. The compounds are crossing into your bloodstream right now, finding receptors that have been waiting for them since before language, finding the precise molecular fit that coevolution built across geological time, doing the work that nothing else can do quite as well, because nothing else is quite as old, quite as tested, quite as shaped by the specific requirements of the living thing you are. There is a word I want to use carefully here, because it can tip too easily into territory that would misrepresent the science. The word is wisdom. Not the wisdom of a mind, not consciousness or intention or anything that requires a thinking thing, but there is something that the ground knows about what the body needs, in the same sense that a river knows the path of least resistance. Not through understanding, through the accumulated result of an incomprehensible number of trials and errors across an incomprehensible amount of time. The soil knows where to send the phosphorus, not because the soil thinks about it, but because the organisms in the soil that were good at sending phosphorus to where it was needed left more descendants. The plant knows what chemistry to make in response to its environment, not because the plant reasons about it, but because the plants that made useful chemistry survived better than the ones that didn't. The human body knows how to absorb and use quercetin from an apple, not because the body studied it, but because the humans whose bodies were better at this were fractionally healthier across their lives and fractionally more likely to survive and have children. All of it is a form of learning without a learner, a form of memory without a mind, a record written in the structure of living things of what worked across time. And what worked across all that time was the conversation, the exchange, the flow of chemistry from soil to root to plant to fruit to gut to cell. The living system maintaining itself by cycling its components through different forms, different organisms, different moments in the great loop of matter and energy that has been running since the first cell closed a membrane around itself in some ancient sea. You are part of that loop. You have always been part of that loop. The apple you ate, if there was an apple, has been finding its way to human hands and human mouths for longer than human civilization. The quercetin in it has been meeting human gut bacteria and human cell receptors for millions of years. The conversation is older than the word for apple, older than the concept of eating for health, older than the first human who paused and thought that this particular fruit seemed to make them feel better. The chemistry knew before the thought did. And right now, in the dark, while the root tips are probing the soil and the fungi are threading their hyphae through the earth, and the bacteria are fixing nitrogen from the air and converting minerals into forms that life can use, all of this continuing quietly beneath and around and through everything. The chemistry of what you ate today is still working, still crossing from gut to blood, still finding its way to cells that recognize it, still doing the thing that it has always done, that it did in the bodies of your ancestors before they had a name for it, that it will do in the bodies of people who come after, in a world that is still growing the things those bodies will need. The conversation does not stop. It does not pause for sleep. It does not need to be understood to continue. The soil works in the dark. The roots grow in the dark. The chemistry moves in the dark. And you, lying in the dark, warm and still, are part of it. Always have been. The potassium in your blood right now. The anthocyanins finding the vessels they were always going to find. The sulforaphane that turned on the machinery that was already there, waiting. The folate doing its careful work in cells that are dividing, quietly, as they have always divided, as life has always divided, making more of itself from the materials the ground provided. All of it older than you, all of it patient, all of it moving through you the way water moves through. Good night.